Hey everyone, it's Miss Lopez. Welcome to your notes on Chapter 6.2, Part 2, the oxidation of pyruvate. We've already covered an overview of basic metabolism, and now we're going to focus in on the breakdown of carbohydrates. Now, we started this process with glycolysis, the breakdown of glucose. At the end of glycolysis, we saw an end product, pyruvate, and we're going to look at what happens to that pyruvate in the presence of oxygen, a process known as aerobic respiration. So let's quickly review where we are going. We have the equation for cell respiration here, and we saw how the process of glycolysis started with glucose. The process of glycolysis broke the glucose down into pyruvate, and we did not see the utilization of oxygen, nor was there any carbon dioxide produced. That is because glucose is only one of 3 steps of glucose oxidation and breakdown. The next step in this reactions or series of reactions, these biochemical pathways, is a process known as the oxidation of pyruvate. So we started with glycolysis, which occurs in the cytoplasm of all cells on Earth. That glucose is broken down into pyruvate. Now, with oxygen present, we're going to move on to the next step, which is the oxidation of that pyruvate. And this is where your mitochondria will take part. So the pyruvate is moved into the mitochondria in eukaryotic cells. When pyruvate is oxidized, it's going to produce CO2, which is carbon dioxide, and acetate. Acetate is then bound to coenzyme A, which is frequently shown as CoA, to form acetyl-CoA. In this process, we see again the key component of NAD+ reduced down to NADH. So here's the reaction. You do not need to memorize the chemical structure of the pyruvate or the acetyl-CoA. Focus instead on the process. So pyruvate is going to be converted into acetyl-CoA. And when that happens, it is oxidized. Now, if pyruvate is being oxidized, something else is going to be reduced. And that's going to be NAD+. So pyruvate is oxidized down to acetyl-CoA. We are going to reduce NAD+ down to NADH. And you'll notice 1, 2, 3 carbons on this pyruvate and 1, 2 carbons on this acetyl-CoA. So to get from 3 to 2, it's going to lose a molecule of carbon dioxide. This entire process is catalyzed by a single reaction, by a single enzyme. Now remember, at the end of glycolysis, we had 2 pyruvate molecules that formed from this 6-carbon molecule. So you've got your 6 carbons here in glucose that get broken down. We're going to harvest that energy, ATP, through a substrate level phosphorylation, and you end up with 2 pyruvate molecules. And each of these pyruvate molecules is going to go through the process of oxidation. So that means for every 1 glucose molecule, you produce 2 pyruvates, meaning that both of these pyruvates will get oxidized down to acetyl-CoA. So you'll end up with 2 molecules of acetyl-CoA, 2 molecules of carbon dioxide, and 2 NAD+ reduced to NADH. So on our bookkeeping slide, let's keep track, or let's add to what we already know from the 1 molecule of glucose. We already had, in glycolysis, 4 ATP produced through substrate level phosphorylation, and the reduction of 2 NAD pluses down to NADH. So from the oxidation of pyruvate to our accounting slide here, we're going to add 2 molecules of NADH. No ATP is produced in the oxidation of pyruvate. We're not going to indicate here the production of the CO2, as that is removed as a waste product and not used in the energy for cell respiration. I hope this video was helpful. Thank you all so much for watching. Bye.